Welcome, everyone. Can everyone come in, grab your lunch, get seated? It's my pleasure to introduce our speaker today. First, I want to say this is a jointly sponsored, well, thanks to the ET Neuro Next seminar series for letting us go off this. This is jointly sponsored through the Howard Hughes Medical Institute, Gilliam program, and the Neural Engineering Center. And it's really my pleasure to introduce Dayo Adewole today. This was, you may remember in the fall, there was an initial visit that was planned, but everything got screwed up with the airlines, which had nothing to do with Dayo. So I don't even need notes really. Dayo has done all of their work at Penn, so it's actually makes it kind of easy. But I do have some notes here. Dayo did their bachelor's in bioengineering at Penn and also mechanical engineering and applied mechanics, and followed by a master's in robotics, where the project was developing an EMG-based control scheme for robotic stroke rehabilitation system, which I think, you know, there's a lot of people who would be very interested in that work as well. I don't think that's going to be the topic today. And the PhD followed by PhD work with, I think, with Casey Collins, is that right? So I think many of us here know Casey. Also with an NSF fellowship, graduate fellowship, in developing the living electrodes that we're going to hear a little bit about today, which this interface that's very exciting between engineering and technology and the tissue, you know, the brain is tissue, in case many of you don't know that. It's soft and squishy, and so we're going to hear about some of that today, but this really complicated interface between technology and biology. Dayo followed with postdoctoral work, currently still at Penn, developing a scalable neuronal aggregate model and test bed for a study of auditory circuits, and really this living electrode kind of idea. So without further ado, I'll turn this over to Dayo. Thanks very much for coming and rescheduling, and we appreciate your time and effort.
Cool. All right. Well, thank you everyone for taking the time. Can everyone hear me okay? Is this good volume? Perfect. So I'd like to thank in particular Garrett. I'd like to thank in particular Oladayo and the departments here at GT and Emory for inviting me to present here. I'm going to be talking about the last several years of research, and it can be summarized within the title. So living electrodes, these optogenetic neural networks or neuronal networks that can be used either for circuit modeling in vitro or for biological neural interfaces in vivo. I recognize that's quite a mouthful, but over the course of the next hour or so, my goal is to break that down into more appetizing and bite-sized chunks for the audience. First, defining what these different terms mean and how these technologies could change what neural interfaces look like in the future. Okay, so I'm a bit of a nerd, and before I knew what a PhD even was, I was introduced to the idea of cyborgs through science fiction. So you've got the $6 Million Man, you've got Steve Austin, you've got the Bionic Woman, you've got the lovable Inspector Gadget, and all of these are fictional representations of an idea that we can combine living things with artificial circuitry or devices. Now, in fiction, this is usually used to grant some measure of superhuman ability. But the people in the audience who qualify currently as actual cyborgs, anyone who's using a pair of eyeglasses, for example, to help with their vision, anyone who's using or taking notes right now to help with cognition, because it's hard to remember everything that you hear. And the many examples in medicine of people who have bravely taken on the risk of implanting these devices within themselves or having these devices implanted to restore some set of function are obviously no less fantastic than superhuman speed or strength or telescoping arms or what have you. So in the real world, people who use prosthetic devices generally get these to restore function after an injury, an amputation, even some sort of disease. And more challenging than creating that mechanical limb to replicate all the degrees of freedom of the natural arm is actually integrating the device like that with the human body. So how do you integrate a device with the body that allows someone to not only control the movement and the actuation of the device, but receive information from the device the same way they would through the natural sensory receptors in the natural limb? So the key to that, and the key to any device that aims to do that, is with a neural interface. So it's a tool that allows for the transfer of information or signals between either the central or peripheral nervous system and an external device. And specifically, these devices are generally communicating with neurons, which, for those unfamiliar in the audience, are the primary functional cell of the nervous system. They communicate generally by sending signals down these long cable-like extensions called axons, outlined here. This is working, if not. And nervous systems themselves use ionic current. So they use the transfer of electrical flow through the movement of ionic species up and down electrochemical gradients versus typical digital devices, which use the flow of electrons to manifest electronic current. So the way neural interfaces generally work is they fall into one of two categories. The first being output or recording, where the idea is that you're harnessing information from the brain or from the nervous system, either the central or peripheral, and using that to affect some sort of device. So generally, you're translating commands into one or more command signals and using those to, for example, move an artificial limb. In the other direction, and one that we'll talk about later in the talk, is for input or stimulation. And with input or stimulation neural interfaces, the idea is that you're delivering electrical current to activate target neurons. And the idea is that you can generate some sort of sensory percept in someone by stimulating the correct neurons in the correct order or with the correct patterns. The go-to example is the cochlear implant, another being the retinal implant, where electrical impulses are used to activate neurons and actually generate either a sensation of hearing for cochlear implants, generally considered the most successful neuroprosthesis to date, or for retinal implants where electrical stimulations allow people to detect some simulation or some simplified version of what we would see with intact organic eyes. So there are, as you can imagine, a lot of challenges that come with these neural interfaces. The first being the incredibly small scale of neuronal signals. These are on the order of microvolts, and it's kind of equivalent to trying to hear someone in the back of the room whisper "hey" to you while you're in the front of the room. One feature of neuronal signals and electrical signals in general is that if you have multiple neurons firing or signaling at the same time, you get this summing effect, which allows you not to detect the individual neuron necessarily, but the summed activity of several neurons at once. And it is this characteristic of neuronal activity, and in fact of electrical activity in general, that allows for a trade-off between the signal resolution or how precisely you can target a specific neuron for input or output and the invasiveness of the neural interface. So at the least invasive and coarsest resolution, you have EEG transmitted through electrodes mounted on the scalp. And diving a little bit deeper, you've got ECOG, so brain-mounted electroencephalographic arrays. And finally, microelectrodes, which actually penetrate the brain tissue itself and can be seen in the form of Elon Musk's Neuralink or the Utah array or BrainGate or many other penetrating microelectrodes that use shanks, needles, or microwires to actually get as close as possible to single cells and can actually provide you with single cell resolution in recording at the trade-off, of course, of having to actually use a surgical procedure to insert those, as well as some other challenges that I'll talk about now. So one of the primary challenges that you'll see in an implantable neural interface is this foreign body response. Now, these electrodes for implantable neural interfaces take the form of stiff probes that are usually inorganic material, and these are amazing at passing electrical current. But the brain, or really any neuronal tissue, is this soft, aqueous, heterogeneous matter. And when it's introduced to this high water content environment, the discrepancy between a rigid inorganic material and a soft aqueous material leads to a set of biochemical responses that we collectively term the foreign body response. Now, this foreign body response has two phases. The first is this acute phase. When you're implanting anything into the brain, you're going to disrupt local blood vessels, you're going to disrupt neural tissue, you're going to disrupt ECM. And this microtrauma, even though it's kind of a ubiquitous nature and inherent to the idea of inserting something into the brain, it still does result in this triggering of local astrocytes and activated microglia or immune cells to the injury site, where they begin to release these reactive oxygen species, these pro-inflammatory molecules, these cytokines, these chemokines, and the goal of this is to actually begin to trigger a wound healing response within the area for this micro injury. Now, over longer timescales, and to be clear, this foreign body response is not a bad thing. If you have a splinter in your finger, you want your body to begin this response to push the splinter out. But when it comes to things that you want to remain within the body, this chronic foreign body response manifests as an ongoing inflammation. And this inflammation, as the brain attempts to deal with the implant, generally leads to either poor neuronal survival around the implant, or the formation of a glial scar, this fibrous tissue that does not contain neurons, but is meant to actually encapsulate the foreign body away from the precious cargo or the neuronal tissue surrounding it. As you can imagine, this foreign body response can negatively impact the ability of an implantable neural interface to actually perform its intended function. So this glial scarring can actually separate the active stimulation or recording sites from the neurons of interest. The local environment is still swimming in these free radicals and these cytokines and chemokines and these activated immune cells. And all of these things can be toxic for the growth and survival of neurons, which we're actually trying to stimulate or record from. At the same time, electrode sites can be corroded. Insulation surrounding the electrode itself can be degraded due to these reactive oxygen species. And ultimately, these serve to reduce the signal quality of the device and can eventually lead to the failure of the device itself. Now, there's an extensive body of research into how and why these brain-machine interfaces tend to fail at the cellular or the tissue level. And these are things that can include device malfunction, device wear, device failure. Not every interface exhibits all of these complications at once, and the extent and the proportion of these really does depend on the material and the mechanical properties of the device. However, several studies, both historic and ongoing, are still teasing out exactly how the foreign body response translates into device failure. But invariably, any interface that you implant, at least with today's current form factors, will result in a foreign body response, chronic inflammation, and essentially a mismatch between the ionic aqueous media of the brain and this rigid inorganic material that is the electrode. So in summary, current implantable neural interfaces are these inorganic devices, and over time, they're going to induce this chronic inflammation and this chronic foreign body response that leads to reduced performance over time. What that means is that their functional timeline, the amount of time over which they're actually useful, is very limited, and this in turn limits how well or how efficiently they translate into medical applications. So what are some strategies to improve neural interfaces given that all of them exhibit or elicit this foreign body response? So the first is really a set of improvement strategies that evolves from mechanical design, so simply making the implants smaller, making them more flexible, providing them with a higher surface area. There are elements of material science involving flexible materials or using nanoscale features to better integrate with the cellular microenvironment. And there's also the introduction of anti-inflammatory molecules to try and reduce that long-term inflammation and in turn help improve the amount of neuronal survival you get surrounding the implant. There's also a class of strategies that's more recent, and these use active biological elements, which I'll explain shortly, in order to decrease the discrepancy between traditional electrodes and the nervous system. And these active biological elements generally take the form of things like different proteins, like laminin, like fibronectin, and these proteins can be associated with neuronal attachment or neuronal growth. The idea is that if you can coat the electrode in these proteins, in these neurotrophins, then you can reduce the amount of inflammation and increase the amount of neuronal survival around the implant for longer periods of time. And these have worked to some extent in a variety of studies. What we've seen generally is that you do get better contact between the neurons and the electrodes. You do have a higher signal to noise ratio over time, and you do see longer biostability or stability of the electrode itself. However, because these are made of predominantly discrete molecules, and these are not produced by the electrode over time, what you tend to see is that once those molecules have been used by the local immune cells or by the local neurons, the effects, the helpful effects of these active biological materials does tend to decrease over time as well. So for these biologically active neural interfaces, we can think of them as existing on a spectrum of biological similarity. That is, how closely it resembles the tissue that it's trying to stimulate or record from. Now you have the uncoated electrode on the far left. This is the stereotypical Utah array or microwire. There have been efforts to coat these with either polymers or different biomaterials, so providing a softer interface that the actual tissue interacts with. And there's also the inclusion of soluble factors or proteins within that coating that are then gradually released from the material in order to reduce inflammation or increase integration with the surrounding microenvironment. There's also the inclusion of living cells, which I'll get into in a little bit. And the inclusion of living cells is motivated by the idea that instead of having a limited supply of biomolecules that is then gradually depleted over time, you have these living cells that can produce these biomolecules and actually provide a more biosimilar interface between the electrode and the host tissue itself, where the tissue is actually interacting with cells that resemble the cells you would see in the brain. And the far end of the spectrum, which has not been presented by any sort of neural interface to date, is the idea that we can use some sort of engineered tissue, so a combination of cells with a defined structure, in order to actually interface with the tissue itself. So the only thing that would penetrate the brain in this far left edge of the spectrum would be organic material, soft material, living cells. So the idea of living cells encapsulated in hydrogels or polymers is not a new one. There have been many groups that have tried to incorporate this concept either for in vitro studies, so incorporating living neurons and support cells into these hydrogels and then studying the neurons themselves, or incorporated neurons into electrode coatings in order to improve the performance of the electrodes after being implanted into the nervous system. There's also been an experimental process of actually polymerizing conductive polymers within the brain itself to form these conductive traces that could be used for stimulation or recording. And all of these have shown a variety of different effects. I think one of the main takeaways from these studies is that while we have been able to demonstrate these cells surviving in vitro, and while there has been extensive characterization of the electrochemical properties of the electrodes after adding these cells and coatings, we still don't have a great idea or a great sense of what the functional benefits are of including these living cells in these coatings is. Does it actually improve the longevity of the electrode stability? These are things that are still open questions and that are still the subject of multiple studies to date. So what is all of this leading to? What are these bioactive electronic interfaces for, and what is this motivating? It's motivating the idea of living electrodes, or these anatomically inspired tissue engineered neural pathways that can themselves be used as the interface between an external device and the brain. And I'll explain what that is, but to do that, I have to give a little bit of context. This isn't research that I pioneered myself. We all stand on the shoulders of giants, and living electrodes in this case stand on the shoulders of something called a microtissue engineered neural network, or a microten. This was pioneered, the idea was conceptualized by Casey Cullen, who works out of the University of Pennsylvania in Philadelphia. And the basic structure of a microten is that you have discrete neuronal populations, so one or more of them, that are connected by the living axon, so those long cable-like projections that neurons use to send information to each other. And all of this is encased for protection and for manipulation within a hydrogel cylinder that we call a microcolumn, microscopic column that's on the order of about 3 times the width of a human hair. Within this microcolumn channel, we also have an extracellular matrix, and that's just to enable axons to grow through that microcolumn, and also provides a bit of trophic support for the neurons just to keep them alive. So this microtissue engineered neural network, or this microten, was originally designed to replace compromised neuronal pathways. The concept was that you could grow these in vitro to the length and the specifications that you need, and use and actually implant it in the brain to replace a pathway that had either been damaged due to injury or disease, or lost or dysfunctional in some way. And so the idea of tissue engineering is not just focused on replacing neural pathways. More broadly, the idea of tissue engineering is designing cells and a structure that those cells exist in to satisfy one of 2 goals. The first would be investigative. So can we model different systems in vitro? Can we study how those systems break down due to disease or injury? Can we test different ways to restore those systems with different types of therapies? And the second goal is primarily medical. Going from investigative to medical allows us to replace the structure of these different neuronal pathways, to restore function in some way, and finally to see if we can include endogenous mechanisms to begin and upregulate regeneration within the body itself. So within neural tissue engineering, in order to tackle either of these aims effectively, it requires the construction and the characterization of the functional architecture of the nervous system. So the nervous system, and specifically the brain, is this complex network of signal processing nodes or groups of neurons that are working together, and signal carriers, or those axons, those cables that carry the information from one collection of neurons to the other. And if you zoom into any particular part of the brain, you can model it as a set of circuit elements where you have some neurons that use specific neurotransmitters to affect other neurons in predictable ways, and those neurons are wired up in specific configurations that you can think of as circuits. So if we have a complex network of nodes and carriers or groups of neurons and axons, it is relatively straightforward to understand how a micro 10 is intended to recreate that architecture at a microscopic level. So this innovation, the idea behind microtens and why they're so significant, is that they actually allow us to distill that functional architecture, localized populations, and long axonal tracts into this self-contained living three-dimensional unit. And it's capable of signal propagation along those internal axonal tracts in the same way that axons carry information within the brain. Moreover, all of this is supported within the soft and minimally invasive biomaterial, and with control over the biomaterial's mechanical and material properties, the introduction of different chemotactic or haptotactic cues within that biomaterial, as well as control over the neurons. So which phenotype we're using to actually seed these structures, the amount or the cell density, as well as protein expression through different genetic transduction or other manipulations. We have a really well-defined tool that allows us to tackle both of these aims of neural tissue engineering, both the investigative or the modeling bases and the clinical or the medical bases. And I'll explain how we intend to do that over the next few slides. So how do we go from microtens to this living electrode concept? And again, the living electrode is the idea that we can use an all biological relay, a fully grown tissue as the go-between for target brain areas and external devices for neuroprosthetics. So the concept is such that we can implant a micro-10, and we can implant it such that with either a unidirectional, so on the right, with a single population of neurons and axons projecting through the microcolumn, or with a bidirectional population, or bidirectional construct that is two populations spanned by long axonal tracts. We could implant this to target a population of neurons within a patient or host. Within that, once the construct is implanted, neurons can form synapses either between the construct and the host and the unidirectional example, or between the host and the construct in the bidirectional example, and allow for the transfer of information between the host and the construct itself. Now, in a surgical plan for this, what you would imagine is that these constructs would be implanted vertically to target specific areas of the brain. What we have here in the bottom right is just an example of the laminar organization of the cortex. And by creating microtens or living electrodes with a specific length, with specific dimensions to target specific layers, we could then use that to target those layers and interface with them by introducing some sort of, for example, photodiode array at the top where the skull is to stimulate the top aggregate, and in doing so stimulate whatever that aggregate is synapsing with. Or in the other direction, if we want to monitor the activity of host neurons by implanting a bidirectional living electrode, the concept is such that activity within the host neurons is reflected in the surface aggregate or the surface population of neurons that can then be detected through traditional means, so through electrical recording or through other optical means by engineering this neuron to fluoresce different types of light when the neurons are active. And I'll get into that and how we prove that that the living electrodes are capable of such later in the talk. So the potential advantages of microtens is in this living electrode concept is one, that we can actually form synapses with the host neuron instead of using electrical stimulation to interface with them. Two, that these living electrodes are encased within the soft biomaterial that could be mechanically tuned to approximate the stiffness of the brain, so minimizing the discrepancy between an implant and the brain tissue itself. By designing these living electrodes to reach a certain length, we can access deeper brain structures from the brain surface thus allowing only organic and living materials to penetrate the brain instead of anything inorganic. And finally, by deciding and specifying which phenotype these neurons are actually, these constructs are seeded with, we can begin to introduce a layer of selectivity based on the neuronal phenotypes. So for example, using glutamatergic or excitatory versus inhibitory neurons to affect a certain neuronal circuit, and by using optogenetics to actually activate or record from these constructs with light as opposed to needing any physical contact methods. And yeah, do you have a question?
Yeah, a quick question. So, with any soft wire or soft material, you have to get it into the brain. So is the plan to use something that's dissolvable and hard but still induce that immediate, stab wound response to get the living electrodes in? Or
Yeah.
do you have another strategy, actually?
That's an amazing question. So I'll talk about implantation methods a little later in the talk, but yes, ultimately, if you're going to introduce something into the brain, unless you're doing away with the needle entirely, you're going to get that acute stab injury. What's significant about these neural interfaces is after the time course of that acute stab injury, there's that ongoing foreign body response. And that's really what we want to tackle with the living electrode concept. So living electrodes' potential significance extends across both arms of tissue engineering, so both investigative and clinical, given further development. So on the investigative side, by developing living electrodes with specific neuronal phenotypes and coupling them together, we can recreate different neuronal circuits in vitro in a three-dimensional microenvironment, so replicating that of the brain, and with control over the phenotype and connectivity patterns of those living electrodes, we can create these large-scale highly biofidelic circuit models that could be used to investigate how those connections form within pathways that are inaccessible normally. On the clinical side, the living electrode concept could be used as a biocompatible signal relay with the brain, one that allows us to achieve high levels of integration with the brain, not just because this is a biologic, but also because neurons naturally form synaptic connections with each other, allowing us to leverage that ability to have a high density of integration between the construct and the brain. And again, by engineering these neurons with specific phenotypes, we have the ability to target the selectivity of these constructs, but for example, by creating constructs with neurons that preferably synapse with a specific subtype of neurons that we're trying to target. So this is a very nice, fancy concept, some very fancy schematics, but what have we actually done to prove this in the real world? So to prove this out, I have three specific aims that I'll be covering today. The first was actually building and characterizing the living electrodes. So how do we build these at all? Can we characterize how they grow? Can we actually control them with light so we don't need to actually touch them with metal electrodes? Two, can we actually show that these living electrodes can form some sort of investigative tool? So can we build living electrodes from different types of neurons? Can we assemble them together to form these larger circuits? And finally, can we demonstrate light-based input-output of these circuit assemblies for study? And finally, for the medical or the clinical aim that we're trying to tackle, can we demonstrate that these living electrodes can be implanted at all? One. Two, do they actually survive the implant, or do they just completely degenerate, go away afterwards? And three, assuming they do survive, is there any sort of early outputs that we can detect as it relates to living electrode survival, as it relates to whether they're growing and forming connections with the brain, and whether there's any function post-implant? So in aim one, we built and characterized these living electrodes. I just want to briefly go over the first generation of the microtens, which had these cell suspensions dissociated with throughout the length of a microcolumn. And these microtens, these first generations, had a random structure, whereas these living electrodes formed with aggregates, these preformed neuronal clusters, actually have a controllable structure because we can form aggregates of a specific size and density before seeding them into the microcolumn. There we go. So how do we fabricate these living electrodes? It's a three-step process. Speaking very broadly, the first is the formation of the agarose microcolumn. This is, like I said, about 3 times the width of a human hair. So to actually generate these microcolumns, we ended up developing a laser cut mold that allows us to insert an acupuncture needle in between the hollows of two semi-cylindrical grooves. And those semi-cylindrical grooves will form the outer diameter of the microcolumn while the needle forms the inner diameter. By then introducing a molten hydrogel, for most of our studies, we use agarose, although other biomaterials are compatible with this process. We can then allow that hydrogel to cool and form the agarose microcolumn with the specified outer diameter, inner diameter, and then cut it to a specific length. Second, we have the formation of the neuronal aggregates. So to do that, we simply 3D printed some pyramids with specific geometries. Within these pyramids, we can then mold PDMS inverts of those pyramids and centrifuge embryonic cell suspensions within those pyramids in order to form these spheroidal clusters or these forced neuronal aggregates that, as you can see in subpanel 5, grow axons in any direction that they're capable of. That subpanel 5 is actually on a 2D surface, so you see this halo of axons growing outward. But in a three-dimensional context, you see neuronal aggregates can grow axons basically from any surface that contacts extracellular matrix. And I'll show some examples of that later. Finally, there's the assembly process where we simply add extracellular matrix to the microcolumn. We seed it with one or more neuronal aggregates, and from there, we simply culture it in vitro. And with this aggregate-based fabrication protocol, we have the ability to create either unidirectional constructs, so featuring a single population, or bidirectional constructs featuring two populations spanned by these long axons. So comparing very briefly first generation to living electrodes, these are just some phase images showing the actual neurons. On the left, you can see that we have these dissociated single cell suspensions, and that over time, these neurons do form these clusters, but the formation of these clusters is completely random. Whereas on the right side, we have these preformed neuronal clusters or aggregates, and we can place these with high precision at one or both ends and just allow the axons to grow throughout the interior of the microcolumn, which you can see through the insets on the bottom right. On the bottom right is an aggregate. Generally, these feature about 8 to 10,000 neurons per aggregate. And on the right, you can see these long axonal tracks spanning the two groups of neurons. Another interesting thing to note is that we can define whether the aggregate is completely internalized to the microcolumn or an internal one, or whether the aggregate is predominantly externalized to the microcolumn. And that's really a function of the aggregate size, which we control through the cell density when we're centrifuging them. So the aggregate-based fabrication method does allow us to create these living electrodes with a living neuronal axonal structure that is highly controllable in terms of both size and density. For unidirectional versus bidirectional living electrodes, we then decided to check whether they actually grew. So we were able to grow them over time, as you can see in vitro, and you can see the projection of these long axons throughout the interior of the microcolumn. You can even see from 3 to 5 days in vitro how after the initial growth period where the aggregates are seeking each other out and forming connections, that there's then a coalescing and a bundling of axons between 3 and 5 days in vitro, where the collection of axons actually separates from the microcolumn walls. This is just another example, about 200 micron scale bar. This is a 6 millimeter living electrode. And more recently, we've been able to grow these up to 1 centimeter in length for longer studies. Our current smallest dimensions, though, are about 350 to 160 microns, or like I said, about 3 times the width of a human hair. And that's a side-by-side comparison just for that. So what about axon growth and survival? Between the unidirectional and bidirectional constructs, we were able to demonstrate that we see a rough scaling of growth rates or peak growth rates that scales with the length of the microcolumn. And we believe this is for a few reasons. I'm happy to go over this towards the discussion period at the end. But one of the primary reasons is that compared to a dissociated culture or a planar culture, these are 3-dimensional microcultures. And so because of that, you have the opportunity for a more biofidelic microenvironment that more closely replicates what you would see in the brain, where obviously our brains are not 2D or flat sections, they are three-dimensional volumes. And the ability to replicate that in vitro, we believe, is one of the reasons why we're seeing these outsized growth rates compared to two-dimensional cultures at the very least. In terms of growth and survival, we were able to stain these with a variety of different proteins, either for labeling or for more functional studies, as I'll get into. This is just a live dead study, or a live dead stain. So we're using calcium AM to stain living cells in green and ethidium homodimer to stain dead cells in red. And you can see there's a paucity of dead cells predominantly within the core of the living electrode, but that the axons themselves look healthy and robust. When we look at survival over time, and we usually look at this as a proportion of living to dead cells, and we look at the survival between 10 and 28 days in vitro, what we're able to see is that although this is an in vitro culture and all in vitro cultures die eventually, the degree of death or the degree of attrition is much smaller in these microtens, in these three-dimensional constructs relative to a two-dimensional planar construct. And again, we believe that part of that is because we're using a three-dimensional environment for the cells to have more realistic, more biofidelic cell-cell interactions that just enable or confer greater survival over time compared to a two-dimensional structure. So this is just another way of showing what we showed in the last slide, where the reduction in viability is significantly lower for microtens of either the unidirectional or the bidirectional variety compared to the reduction in viability for a 2D culture, which approaches about 50%. Looking at network structure and function, first I'll get into the physical structure of these living structures. This is an M-Cherry and GFP stain. So this is nothing but a fluorescent label just to look at the neuronal cell bodies and axons. And what we were able to show with these labelings is that aggregate neurons do project these long axons towards the opposing aggregate. And as we'll see in more functional studies, the activity in one aggregate and the other aggregate is connected, as we can see over time. But this is just an example of the structural integration that we were able to see by 10 days in vitro for this was a 3 millimeter bipolar living electrode. Okay, getting into more functional characterization, we were able to label these living electrodes with different calcium reporters like GCAMP. And once we're able to do that, we can look at the different populations of active neurons and begin to run some statistical analyses to say whether one or both aggregates is functionally connected, both within itself and to the other aggregate in the case of bipolar constructs. So the calcium signals within a bidirectional living electrode do actually suggest that there's functional connectivity between the aggregates. And before I get into how we showed that, I just wanted to show briefly what our analysis looks like. After we label these with GCAMP, we outline specific regions of the living electrode and then look at the fluorescent traces and run statistical analyses on these traces to look at functional connectivity between the different regions. In the case of a functional study looking at the connectivity between aggregates, what we were able to do is take those traces and generate some correlation matrices where we're looking at both connectivity within an aggregate, as evidenced in the highlighted region, between aggregate 1 and aggregate 2, as well as the connectivity between aggregates, or this highlighted region. And you can see that there's clearly a higher degree of connectivity within an aggregate versus between aggregates. And the reasons for that, we're still trying to figure out, but we have some early ideas, which I'm happy to talk about later in the discussion. So we've shown that these constructs can be built. We've shown that these constructs are functionally and structurally connected. The next step for this aim was actually developing an ability to control and monitor these with light. So by transducing one aggregate with an optogenetic actuator or an opsin, in this case channelrhodopsin, and the other aggregate with an optogenetic sensor, in this case RCAMP, culturing the aggregates within a single living electrode, we could then stimulate one side while recording from the other, all with light. So we're not touching this with any sort of electrodes. We're simply stimulating with light, shining a specific intensity and wavelength of light on one side, and then looking at the network activity as measured through RCAMP in the other. So this is an example of one such construct. So this is just the fluorescent microscopy image of the same. And by performing a similar analysis, we can take recordings of the output aggregate featuring RCamp. We can take different regions of interest within that aggregate, and then we can overlay these with the actual time course of stimulation for each experiment to determine, does the stimulation in one aggregate actually reflect the activity in another aggregate? In other words, is there a functional synapse between one aggregate and the other that we can actually trigger with light? What we were able to show through a host of experiments is that there is a time-locked effect. That is, there is activity in the output aggregate that does seem to reflect the time course of stimulation in the input aggregate, and that in the absence of that stimulation, we do not see the same response in the output aggregate. We were also able to show that by increasing the intensity of light, we also saw an increase in the fluorescent signal, which could reflect an increased recruitment of the input aggregate neurons as we increase the intensity of light, which would then result in activating more of the output aggregate neurons. Also, crucially, we were able to show that by shining a different wavelength of light at the same high intensity on the input aggregate, we did not see a response in the output aggregate, indicating that this was not just a photoelectric effect and a bunch of wishful thinking, but that this is actually an effect due to a functional synapse between one aggregate and the other. So summarizing that first initial research aim, we have these newly developed living electrodes featuring these forest neuronal aggregates. They have an incredible capacity for growth. They have a capacity for survival that outstrips 2D cultures, and they form this functional architecture harking back to the functional architecture of the nervous system with these discrete neuronal populations connected by these long axonal tracts. And we can engineer these for all optical or light-only input and output, or control and monitoring. So going into the second research aim, we wanted to demonstrate that we could build these out as an investigative tool, building with the two aims of neural tissue engineering. And the goal here was to build these with distinct neuronal subtypes, assemble chains of these living electrodes into multi-unit assemblies, and then demonstrate light-based input-output of those assemblies. So distinct neuronal populations in this case refers to whether we can build them with the glutamatergic or predominantly excitatory neurons, or GABAergic or predominantly inhibitory neurons. This is just an example of the same. We're able to take neurons harvested from primary rodent cortex or from the medial ganglionic eminence, which is predominantly inhibitory at that particular time point. We're also able to show that we could make these with dopaminergic and hippocampal neurons, which is outside the scope of my specific research, but that is an ongoing arm of research in Casey Cullen's lab. We're also able to show that by connecting these, they naturally form these multi-unit assemblies, and that's just a matter of synapses forming between the two constructs as the neurons grow. We were able to form a few different configurations, mostly just to see if we could. But the idea here is that once we connect these together, we can begin to stimulate and record from them with light to begin to understand how these connections are forming and whether there's any structural features that relate to or that can be correlated with the functional outcomes that we see. So this is just looking at, again, GCamp within a cortical neuronal assembly, as well as an overlay featuring traces from the indicated regions of interest. One interesting thing you can see here is that it looks as though the blue region of interest, the blue region of interest does seem to have a slight lead in its peak fluorescence compared to the other regions of interest. Now, whether that means that the blue aggregate is driving the others requires more statistical testing, but it's just an example of some of the questions that we can begin to generate as we build and analyze these constructs. Here's another example of a hippocampal assembly, and you can see that the activation pattern is different, and we are currently attributing that to the different phenotypes in predominantly cortical versus predominantly hippocampal neuronal isolations. But as you can see, whereas in the cortical neuronal assembly, one seemed to be leading the other, these all seem to have about the same time course of activation and quiescence, but that it's a sustained period of activation when that's the case. And I'll just let this play just to demonstrate what that looks like.
Doing here to drive this fluorescence?
Nothing. So this is the construct's spontaneous activity without any external stimulation. Okay. So in AIM 2, we were able to show that these aggregate-based living electrodes could be built with multiple distinct neuronal subtypes, can be coupled into multi-unit assemblies with these various topologies. And finally, we were able to demonstrate that there are fundamentally different characteristics and activity based on the subtype that we use to build these multi-unit assemblies. One set of studies that has not been completed is whether we can actually begin to stimulate different aggregates and look at the corresponding response in the entire construct. Yes.
You push the neurospheres into certain differentiation paths?
No, so these were predominantly harvested. This was a primary neuronal source, so terminally or nearly terminally differentiated from rodent embryos. So we were able to build these. We were able to put them together in kind of a Lego building block formation. Finally, we want to look at the clinical aim of neural tissue engineering and see whether these could actually be implanted, whether we can implant them without destroying them on the way in, and whether they survive that implantation process. So to do that, I developed this implantation method, which just consists of a PDMS collar, a glass slide, and a little bit of cyanoacrylate glue. This is an example of a living electrode post-implant into a brain phantom, just to look at the signal from the axons and the cell bodies post-implant. There is a bit of artifact here, but we can see that the axons, for the most part, seem to survive that implantation process. And when we looked at this under a 2-photon microscope after actually implanting it into a rodent cortex, we're able to localize the surface level aggregate, given that the bottommost aggregate is deep within the brain. And we were also able to use the same 2-photon to look at individual neurons and cancel out the motion artifact due to breathing in order to identify whether the activity within the neurons reflected activity that we saw prior to implant. So we did just that. We took some calcium imaging of the implant of the living electrode after implantation. We took some regions of interest corresponding to cells within the topmost aggregate, and we were able to then take some traces, run some signal analyses, and we're able to identify the motion due to breathing artifact, as well as activity bands that seem to be in about I believe that's the 1.5-ish range, as well as in the 0.25 range, or the 0.25 hertz range, sorry, for activity within the neurons themselves. Now, what this means, we're not entirely sure, but what we do know is that this 1.5 hertz activity band is the same band we saw within the constructs prior to implant. So whatever's going on, we see that the network activity is maintained at least out to 1 month after we implant these, which at least to us indicates that the structure is maintained enough to do that. Looking at survival and outgrowth. So we did some immunohistological staining for GFP, which would allow us to identify the living electrode neurons, IBA1, as well as GFAP, which allow us to look at activated microglia, as well as astrocytes within the area. And while this set of studies is still ongoing and we don't have any strong conclusions from this, we do see a moderate immune response at 1 month post-implant. And we're still looking at what the implications of this are compared to, for example, a micro stab wound or the implantation of a traditional microelectrode. Basically, we want to compare to see, is this about the same amount of immunoreactivity as you would get with those other implants, or is this less, or is this more? And that will determine our next steps for any in vivo studies looking at this. This is just another example of the same, as well as a stain for synapsin, which we use to determine whether any synapses were forming between the living electrode and the brain, at least at 1 month post-implant. And looking at some zoom-ins, we were able to co-localize GFP. So again, looking at living electrode neurons, as well as synapsin. So looking at the presynaptic marker for a synapse, and co-localize a few as indicated by the white arrows. Again, this is not a conclusive study by any means. This is a proof of concept, but the goal with this is to show that post-implantation, these living electrodes are not only alive, they're not only functioning, but they actually form functional synapses with the brain. And this is, again, something that's ongoing. So summarizing aim 3, for aggregate-based living electrodes, we were able to implant them into rodent cortex along with a customized cranial window. We saw that they maintained the structure that they had following implant, and we were able to provide not only functional optical readout of the living electrode post-implant, but early evidence that at least enough of the structure remains that the living electrode may still be functional at least out to 1 month, with further studies ongoing. So summarizing all of this, a lot of medical technologies do rely on neural interfaces. There are some newer technologies that could rely on neural interfaces, but currently neural interfaces do have this inflammatory response, this foreign body reaction, and it's important that we address that however we can. Living electrodes are just one way that we're looking at to try and do that, but it's one way that we think has a lot of different benefits depending on the application you use them for. Again, these can be used either as an investigative platform for modeling different neuronal pathways in vitro, or as a synaptically mediated biological neural interface in vivo, given further development and the formation of synapses between the construct and the brain. We were able to show that we built and characterized these aggregate-based living electrodes, that they grow really rapidly, that the neurons survive out to at least 28 days, and we have some unpublished studies looking at 40 days in vitro for a live 40 days in vitro viability with promising results. We're looking at functional networks. So can we control these with light? Can we monitor them with light? And what do those light-based outputs mean in terms of the structural connectivity of the aggregates and the living electrodes? We're able to build and monitor living electrodes from distinct neuronal subtypes, as well as assemble them into multi-unit chains, talking towards the investigative aim. And we were able to demonstrate a proof of concept implant in rodent cortex. And within rodent cortex, we implanted these in V1, just for your information. And we were able to show that not only do they survive and grow post-implant, but that there is early evidence of their continued function out to at least 1 month, as demonstrated by in vivo calcium imaging. So as with any new technology, there's a whole host of challenges, a lot of questions, and some potential future directions. The first is, can we make these even smaller? You know, we have metal microelectrodes that are on the orders of microns. 300 microns is small, but we think that we can get smaller. Can we optimize our delivery? So one delivery platform that we've looked at is using a polymer that is stiff when it's dry, but upon contact with water softens. And by forming the actual microcolumn from that, we were able to effect needleless microinjections, to your earlier point, where the microcolumn itself serves as the force, provides the force necessary to penetrate the brain and then softens to the level of brain stiffness. We're still looking at histology for these implants to look at the amount of outgrowth we're getting, as well as the amount of ingrowth we may be getting from host neurons into the construct, as well as the amount of synaptic integration, synaptogenesis, and synaptic integration we have. And we're looking at a host of other things, including how can we better select for different phenotypes for specific neuromodulatory effects? Are we able to control the synaptogenesis of these post-implant? And can we actually characterize the function of these neurons using calcium imaging and correlate those with different structural information so that we can actually identify a living electrode that will do what we want versus a living electrode that is non-viable, as would be necessary if you wanted to implant this in someone's brain. Finally, we're looking at some large animal and human scale constructs, so using human iPSC-derived neurons to generate these in lab, and also looking at some larger constructs. So like I said, we're making these about 1 centimeter long now and looking at how much longer we can get these to grow. So that's living electrodes in about an hour. This was funded by the NIH as well as the NSF. And of course, could not have been performed without a whole host of people, most of whom are not here today, but many of whom contributed to the success of the research so far, and many of whom continue to today. There's a host of publications if you'd like to learn more about this on your own time, but that is my Living Electrodes talk, and I'm happy to take any questions at this time.
Yeah, we do have time for questions.
I don't know if you've looked into sniffers at all, but for a while, there's been this idea that you can put optical sensors and aggregates of cultured cells and implant them into the brain and use the optical readout to read out things like dopamines. This was before we had, good GPCR-based sensors. And a problem that I think a lot of people ran into was they got recognized effectively as a tumor because you're implanting a big aggregate of cells in the brain where aggregates normally don't show up all of a sudden. So, is there any thinking about whether it makes sense to be implanting aggregates of cells and whether or not, like your problem is kind of harder because if it gets recognized as a bad biological thing, that's almost worse than, a foreign body response.
Yeah.
Right. So I don't know if you have any thinking about how to avoid those types of problems with implanting biological matter.
Yeah. Yeah, that's an amazing question and something that we think about. One of the first barriers to that kind of immune response is the microcolumn itself. So we can tune the stiffness of it to degrade over the course of weeks or over the course of months, and that we believe is going to serve as a physical barrier to any immune cells that might recognize and then try to attack the precious cargo, which is really the axons, but also the neurons within. Yeah.
Curious about some controls for the synaptic connectivity testing. So for example, you see a change in the response in time as they grow towards each other, or have you considered using electrodes to stimulate one side so that you wouldn't risk having an optical signal pass through the emission filter on just try to maybe explain your controls, make sure that these things really are connected.
Yeah, absolutely. So we've done a few different controls. I didn't go over them in detail, but we've done the electrical stimulation of one. We've also cut the axon traces after the fact and then stimulated with light to see whether we see any response. There's the stimulation with the off-target wavelength, and there is one more control that I'm blanking on. But yeah, we think about that optical pass-through potential a lot, and how we could mitigate that when we're running these experiments.
I have probably in the naive question. Great talk, by the way. Super excited to see how your work has evolved over the years. In the case of the unipolar living electrode, what is it growing towards? And then when you implant it, what does that mean for growth?
Yeah, great question. So right now, the unipolar living electrodes aren't growing towards anything. We see the axons reach the end of the microcolumn, and if we don't do anything with that living electrode, we do see the axons start to degenerate and die back because they're not connected to anything. What that means functionally is that once they've reached a mature point, we have a limited time window to actually place them next to something that the axons would want to form synapses with, but we don't know what that time window is specifically. Yeah, in terms of what that means for when we implant it, it means two things. One, that we have to protect the axon terminals as we actually penetrate the brain, which we figured out a way to do with some fancy engineering. And the second is that we believe that the unipolar constructs may provide some benefits over the bipolar constructs for the amount of integration we get. That's something that we think may be the case, but we don't have anything to prove that yet.
Yeah, there's a couple questions. Bilal Peter asks, why is the timescale of correlation between aggregates rhythmic? Does it go away with blockade of synaptic transmission? And then as a follow-up to that, are the vote responses TTX?
Got it. So why they are rhythmic? Still open questions, but we did perform a blocking study and we did see the blunting of that activity. So we think that's just another form of confidence that there's something going on between the aggregates that allows them to have that kind of ping-ponging activity, because the addition of TTX just blocks that completely. Why that's the case, still unsure.
Maybe one more quick one for the optical readout in vivo. I think it's super provocative. Do you have a control to suggest that it's reading out from the brain and these aren't cells just communicating with each other up top?
No, so that's something that we don't know yet. It could be, yeah, it could be either one. At this point, we don't know. And at 1 month, I would say it's probably more so them communicating with each other. But yeah.
I have a question. So, this is maybe a little bit a crazy question, but there are groups that are working on ways to use cells, bioluminescence to in turn stimulate and make them, with options to make them sort of self-stimulating in some ways. Is that kind of idea on the table for something like this, do you think?
It's definitely on the table, but not something that we've explicitly discussed. Yeah, that's some really cool technology. Yeah.
Well, thank you very much for coming.
Thank you all.
Very cool work. Has to be over.
Oh, good.
Kids are back at school this week, so, and I got all my study section, so. On time, I know, but it's, I just don't know any better, right? That was on NH.
For the what? Oh, yeah, so that's downstairs.
Okay.
Yeah, well, there's an elevator in that part of the building, and you just take it up.
You suggest bringing it up?
It's like ideas, it's a crazy idea.
Like, I brought it. You would think one's graduating back.
Trying to.
Something.
That's fine.
Yeah, I just need a.
So I can-
So I
I think we should say,
So I probably.
oh, I think I'm just gonna, I'm gonna walk in like.
PhD comics about that. Did you know this comic strip?
So that's great. I can tell you about either you told me this or.
But there was like, how long you're. How long you think it's gonna take? How long-
Very clear, right?
There are no calculations.
Yeah. So that's where I'm at. I feel like, and I just feel like I just need to commit to spending just like
I know.
long
Well, do it
days. Yeah.
Or the ones that get us, and like, there's besides us.
Right, yeah.
Student group.
No, I don't. Cool.
All right.
Get back to it.